I probably won't go through all the slides. I'll start. So I'm Wilson Wong, professor of biomedical engineering. Um, so I'm very interested in cell and gene therapy, especially immune cell therapy, CAR T cell therapy. Just a quick recap. You can now take patients' T cells out of the body, use genetic engineering, introduce a chimeric antigen receptor, which is a tumor targeting receptor, into the T cells, so it will express it. And then usually there's an antibody on the surface of that receptor. And what it does is that it'll allow the T cell to find the tumor, expressing a specific biomarker, HER2, for example, or CD19, and then it'll activate a T cell, and the T cell will kill the tumor. Now, so my lab has always been very interested in finding ways to improve the safety and specificity, and lately we're very interested in reducing the cost of this type of therapy. So one thing about these CAR T cell therapy is that it could be really toxic in a way that it can be super activated when it encounters a lot of cancer cells. So over the years, we've been developing another safety switch that allows you to, in addition for using the biomarker to activate the T cell, we also develop a way that you can use a FDA-approved drug such as Grazopavir, which is a hepatitis C drug, to control the activity of that CAR. So you can turn it off when you need to by using the drug. Another problem we've seen a lot in these therapy, not just in CAR T therapy, but also in a lot of cancer therapy, is that it's not specific enough to the tumor. So what's nice about using cell-based therapy is that we can perform rudimentary logic to improve the specificity. So in this case right here, you can see it right there. For example, let's say the tumor cell has this red biomarker and your CAR T cell can kill it. Now, you can imagine a scenario where the healthy cells that you're trying to protect also have the red biomarker, but also has a black biomarker as well. And we can engineer a circuitry that allows it to, so that it will not attack the healthy cells, because the black biomarker is actually inhibiting the CAR T cell, only attacks the tumor. So I'm very proud to share that this project is actually a collaboration with Senti Biosciences. It's already in phase 1 clinical trial for AML, and it has a really good data so far. It's very early. 2 out of 3 patients have complete remission for AML, so I'm very proud of that. And then finally, we've been also working with Professor Grindstaff and Florian Duan right here at the medical school. We've been figuring out ways to improve RNA therapies beyond vaccines. So a lot of us have the vaccine, mRNA vaccines. We've been working with the self-amplifying versions of that so that it lasts longer. And then what that does is that it will also open up as a gene delivery platform. So one thing that we are very interested in making CAR T cell using RNA, as opposed to, traditionally we make them with lentivirus. And then also we do them in situ, meaning that we don't have to take patients' T cells out and engineer them anymore. We make CAR T cells directly inside the patients using the vaccine technology that we've been making. So hopefully it will dramatically lower the cost as well. So thank you very much. I'll stop here.